My name is Colleen Poulton. I am a student at Boise State University. I decided to do my project on the field of genetic engineering. But what exactly is genetic engineering? Genetic engineering consists of manipulating or modifying genetic information by either introducing genes from outer organisms or modifying an organism's own genes. We've been discussing the manipulation of DNA through genetic engineering, but what exactly is DNA?
DNA is the instruction manual for how to build life. From animals to plants to humans, it defines us all. Information is stored in DNA using 4 types of molecules, which come in pairs. We have billions of these pairs.
These pairs make up what are called genes. But what exactly are genes?
A gene is a special stretch of DNA, a sequence of A's, C's, T's, and G's that code for something. A gene contains information for a cell to read and use, but what exactly does that information do? Genes make proteins. Those proteins then interact with each other and all sorts of chemicals inside the body to build things like eye pigments, freckles, and mood-altering hormones. A single strand of DNA contains thousands of genes or unique protein recipes. Humans have roughly 20,000 altogether. Some genes are small, only about 300 letters long. Others are well over 1 million. The length and sequence of a gene determine the size and shape of the protein it builds. The size and shape of the protein determine the function that protein will have inside the body. Because all creatures use DNA in pretty much the same way, genetic engineers have found that if they take a gene from, say, a bacteria cell and insert it into the DNA of an animal or a plant cell, that animal or plant cell will then read the new gene and produce the bacterial protein.
After learning about the amazing field of genetic engineering, I wanted to discover more about how this process could be applied. To achieve this, I conducted an interview with Professor Julia Oxford, an expert in cartilage regeneration. Professor Oxford, in her interview, discusses the concept of stem cells.
One exciting approach is to take those induced pluripotent stem cells, or maybe they're even patient-specific adult stem cells, and put those with a decellularized scaffold. So make that combination. And then that should give you patient-specific cells with the right environmental cues, and maybe that's our best chance for healing.
After illustrating what stem cells can do, you're probably wondering, what are stem cells? Stem cells are basically blank slates of the cell world. They are said to be undifferentiated, meaning unlike skin cells, which have special genetic instructions telling them to be skin cells, stem cells have no instructions. Stem cell research is so exciting because now through genetic engineering techniques, scientists can give stem cells special instructions to develop into bone cells, cartilage cells, blood cells. The options are limitless. This revolutionizes medical science because now the question isn't what can we do, it's what can't we do.